In the next few sections for the endocrine system chapter, we're going to consider particular examples of endocrine function, starting with looking at the endocrine function of the pancreas. The pancreas is often referred to in terms of endocrine function. However, it's actually not primarily an endocrine gland. Its endocrine function is very important and very well documented in clinical cases, and probably many people are aware of it through diabetes. But with the other glands we usually talk about in the endocrine system, this one just isn't primarily endocrine in function. The pancreas is located just posterior to the stomach, which has been removed in this representation so we can see the pancreas. And it sits in sort of the crook of the duodenum, the initial portion of the small intestine. It connects to the duodenum. The pancreatic duct, the green line that we see running through the gland, is open into the duodenum where it releases its digestive enzymes. And that's what the pancreas is really responsible for, is producing a lot of digestive enzymes that the small intestine is going to use, as well as other components of digestive juice. The endocrine function of the pancreas is minor as far as the amount that we're talking about here, amount of its function. As you can see in the picture, scattered through the pancreas are these little, florets of tissue-looking things, which are called pancreatic eyelids. Now, islet means little island, so these are little regions within the larger digestive gland performing the endocrine function of the pancreas. Now, in the inset that we can see off to the side of the pancreatic islet, the staining quality of the islet cells, the endocrine tissue, is lighter than the surrounding exocrine area, or the acini cells that are responsible for making the digestive enzymes and other components of juice. I want to correct what seems to be a mislabeling in this figure. It's not necessarily incorrect, but generally speaking, a class of cells called beta cells are going to be in the center of the islet, while as a different class called alpha cells are going to be at the edge. That's not 100% the case, so this particular one could actually be labeled technically correct, but more often you're going to see beta cells in the central region. I doubt that this particular inset is labeling the cells accurately, because with this type of staining, just a basic cell stain, you can't really tell the difference between beta and alpha cells. You need a specialized stain to highlight the differences. So the labeling here might be off just because there's not really enough information to say one versus the other. But the rule of thumb that beta cells are in the center and alpha cells are at the edge is an acceptable way to kind of think about them, especially when we're dealing with cell stains like we do in this course. To really talk about the endocrine function of the pancreas, however, we want to consider the homeostatic mechanism behind regulating blood sugar levels or blood glucose levels. And so let's consider the finer points of this figure. To begin with, we want to consider where the homeostatic loop starts. Now, actually, this is two loops, so more of a figure 8 configuration here as we look at how blood glucose levels go down and how they go up. Now, I really like this image. It starts by introducing the idea of the homeostatic set point, the variable that we're interested in, represented with a blood sugar level testing apparatus. So a diabetic would in fact do this to measure their own blood sugar levels with a small finger stab, prick, whatever you want to call it. A drop of blood is taken up into a collection device, which is read by a little sensor and can establish the particular level of glucose in the blood. Now, the range for normal levels is 70 to 110 milligrams per deciliter, and in this figure they just choose 90 as a nice central number of that. The milligrams per deciliter unit that we're seeing here, that's milligrams of glucose in a deciliter, or 1/10 of a liter of blood. And that's just a standard unit used to look at concentrations of things in blood. So to begin, let's consider the imbalance in the homeostatic mechanism when blood sugar levels go down, which is called hypoglycemia. Hypo for below, glyce for sugar, eme referring to blood. So low blood sugar levels. The number that we're given here is down to 50 milligrams per deciliter, which is definitely below the range at the start of the homeostatic loop. When this happens, alpha cells in the pancreatic islet release glucagon, and that gets into the bloodstream, travels throughout the whole body, and affects its particular target cells, those cells with the glucagon receptor on them. The primary targets of glucagon are cells of the liver. The liver is responsible for two responses that will increase blood glucose levels. First is glycogenolysis, which is breaking down glycogen to release individual glucose molecules into the blood. Glycogen is just a starch-like molecule that's chains of glucose molecules stored for future use. It also stimulates gluconeogenesis, which is essentially a reverse of the glycolysis reactions necessary to initiate production of energy-rich molecules from glucose. The remnants of glycolysis are 3-carbon molecules such as pyruvic acid, and gluconeogenesis takes those and returns them to glucose structure. It can also use some other sources of carbon to get there. It's not really an energy-efficient way of doing it, but the liver is using energy to reconstitute glucose so that glucose is available for other tissues of the body to use. So it is going to eventually help, although it's not very efficient. The book also mentions that glucagon inhibits body cells from taking up glucose. That's probably not really the clearest statement. Glucagon's acting on functions which insulin, which we'll look at in a second, counteracts. And so glucagon, to some extent, can counteract insulin's functions, except that glucagon does not really target all body cells. Primarily it targets the liver tissue. There are other potential targets of glucagon, but not really all body cells, like we'll see is true for insulin. And by releasing glucose molecules from the liver into the blood, glucagon returns blood glucose levels to their homeostatic range. And so we're closing out that first part of the loop. Then we see an increase of blood glucose levels from the homeostatic range into hyperglycemia. And the number given here is 160 milligrams per deciliter, which is certainly indicative of that. We would tend to see hyperglycemia just after eating food. As we absorb nutrients from the food that we've eaten, they get into the bloodstream and the levels go up. Then, in response to that, the beta cells in the pancreas start to release insulin, which gets into the blood supply, goes throughout the body, and has effects on its target cells. The target cells for insulin really are all cells of the body, with a few exceptions. For example, the dead cells in the epidermis, the outer layers of the skin, are not going to have insulin receptors on them because they're not living, they don't need energy, they're not going to take it up. But otherwise, pretty much all the cells in the body have insulin receptors on them because they all need to take up glucose from the blood when it's available. So primarily, insulin causes the uptake of glucose from the body so that cells can use it in driving cellular respiration reactions, making ATP. It also tells the liver to stop releasing glucose by breaking down glycogen and to stop making more glucose through gluconeogenesis, neither of which is needed. And in stopping the process, especially of glycogenolysis, it's going to also tip the liver over to storing that glucose back in glycogen in the liver. There are a few other effects that insulin has that are not pointed out in this figure because they're beyond the straight homeostatic mechanism that we're talking about here. When you eat food, of course, your blood glucose levels are going to go up, but also amino acid and lipid levels are going to go up in your blood as well. You've just eaten food, and really all three nutrient molecules are going to be present. Insulin, while it's helping to regulate blood sugar levels, is also used to stimulate the uptake of other nutrient molecules. It's not directly about regulating those levels in the blood, but rather just the opportunity to use one signal for multiple functions together. It's just an efficient use of all of that. Also, along with the production of glycogen in the liver, insulin is going to stimulate more production of glycogen in muscle cells. And by all of the glucose being taken up into the cells of your body, this again reduces blood glucose levels and gets us back to the homeostatic set point and closes out the second part of the loop. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. The beta cells are responsible for secreting insulin. Alpha cells are responsible for secreting glucagon. There's also a delta cell and a gamma cell, which we aren't discussing in this course, but they also play a part. And then the acinar cell, or the acini cell, is the exocrine cell that's secreting digestive juices.